Alright guys, the goal of the videos in this series is to go through cellular respiration, which includes glycolysis and pyruvate oxidation and citric acid cycle or Krebs cycle, same thing. Electron transport chain, we're going to talk about the aerobic side of things, anaerobic fermentation. So there'll be a series of videos putting this all together. In this video, we're trying to get the big picture. So before you dive into the details, it really makes sense to understand the major concept of what's going on. All we're trying to do is extract energy from molecules in food. And so the way this is typically done is most classes and textbooks use glucose as their example, right? C6H12O6 is glucose. That is our starting material for glycolysis. So remember, you can also use amino acids from protein or fats like triglycerides to put energy into the system. And we'll mention those a little bit later. But for right now, just use glucose as an example. You're taking in glucose and oxygen and the products of this entire reaction. So if you start with glucose in the beginning, go all the way through glycolysis, go into mitochondria, go through Krebs or citric acid cycle, go through an electron transport chain. At the end of it, the net effect is you breathe out CO2 and you breathe/pee out a combination of water along with that CO2. And so in the process, all we're really trying to do is extract energy, right? So you can think of energy as one of the products of this entire process. That's the only thing you really want to keep, right? You don't care about keeping the actual mass of glucose, right? The carbon, the oxygen, the hydrogen, it can all go. CO2 and water. The energy is what we want to keep. And so all of these reactions, it's basically just saying we're taking this big reaction, right? Glucose plus oxygen gives CO2 and water, and we're breaking it apart into a bunch of little steps, right? And so each of these individual steps, which depending on the level of class you're in, you may or may not have to know, but probably not. So glucose to glucose 6-phosphate to fructose 6-phosphate, these are just baby steps in the process of breaking apart that glucose and extracting the energy from it. So by the end of all of this, we can take the CO2, right, that are waste products, breathe it out, get rid of it, along with water that's also produced, right? So by the time you get to CO2 and water, those are low energy molecules, right? We don't have any use for them anymore. Right? You don't want to just keep accumulating CO2 and water, right? And the energy in the process is getting converted into ATP, right? So that's our whole goal. Now, in order to do that, this all comes down to some fundamental chemistry. So first concept that's critical for this, you're going to hear a lot about redox reactions. The word redox really just means a combination of reduction and oxidation, and they have to go together. So if something is being oxidized, something else has to be reduced. And when it comes down to it, oxidation just means the molecule or atom is losing electrons, and reduction just means we're gaining electrons. Okay? Electrons don't disappear, and so if you're going to oxidize something and take away its electrons, something else has to be reduced. Now, this can be a straight up, like, complete loss of an electron, or it can be partial. And so in this example, they're showing you oxidation of methane, and the thing that's happening here is, remember, oxygen atoms are very electronegative, and so in methane, carbon gets to keep its electrons for the most part, because its electronegativity is similar to hydrogen. Remember, in chemistry, electronegativity just means how attracted something is to electrons. So carbon and hydrogen are sharing their electrons pretty evenly. As you start adding oxygens, those oxygens pull the electrons away from carbon, right? And this O2 with a double bond pulls electrons away even more. We start adding more oxygens, carbon's losing more and more and more electrons until we finally get to this CO2 where there's a very, very strong pull, and the oxygens wind up pulling those electrons away from the carbon so much that this is a very, very oxidized state, right? Carbon hasn't completely lost the electrons, but in these bonds with oxygen, the electrons are shifted towards the oxygen so much that we say it's in a much more oxidized state, right? This methane at the beginning is very similar to glucose, right? Glucose has a lot of carbon-hydrogen bonds. That's where the energy is, right? And so typically when we're over here in the reduced state, there's higher energy. That's why you can burn methane, right, and get a flame out of it. It's combustible because it's high energy. Carbon dioxide doesn't burn because at that point the bonds are relatively low energy. There's not a whole lot left to extract out of it. Okay, so we can reduce things by pulling the electrons away from the carbon, from the atom. You can also oxidize and reduce by actually moving entire electrons around, and that's when you're going to see plus and minus charges. So this NADH molecule is a very, very important electron carrier in this entire process. It's going to be a product of glycolysis and of citric acid or Krebs cycle. So when you see NAD+, NAD+ is a reactant for glycolysis and Krebs cycle, meaning you need it to start off. And we say NAD+ because this NAD molecule has a single positive charge on it at that nitrogen. Probably don't have to memorize exactly where it is, but the point is, if we add an electron to NAD+, you get a cancellation of that charge. The negative electron cancels out the positive charge. Now we're neutral. And so you would say that NAD+ in this case is being reduced, right? Because gaining electrons means reduction. If you broke that electron back off and went from NADH back to NAD+, that would be oxidation, right? This NADH molecule is just going back and forth between being oxidized, reduce, oxidized, reduce. And so when we take in high energy molecules like glucose, we are oxidizing that glucose and using its electrons to reduce NAD+ into NADH, right? And then later on, that NADH is going to hand the electrons back off again to the electron transport chain, right? That'll come in a later video. But remember, the whole purpose is we're just shuffling electrons, right? And as long as you remember that typically the reduced form is generally high energy, and as you go to the oxidized form, these molecules are relatively low energy, which means that along the way, you had to have energy released, right? And that energy that's released is eventually what's going to make us some ATP. That's the whole goal of this entire process, right? So linking that with ATP production, right? And ATP molecules, remember the energy is stored in the bonds between these phosphates. The negative charges on those phosphate groups give ATP properties where its bonds that hold the phosphates together require a lot of energy to be broken, right? And so when we're talking about oxidation and reduction reactions, when we oxidize something like glucose and extract the energy out, we're using that energy to form those phosphate bonds between ATP. That way, later on when the cell's doing things like running enzymes and muscle contractions, we can use that energy that's now stored in the ATP bonds to get things done, okay? So this entire cycle, when we say that we're using or making ATP, it really is as simple as breaking off and then reattaching that third phosphate, right? ATP is adenosine triphosphate. The tri is telling you that there are 3 of them, and these are high energy bonds. So that just means if you break them, the energy is going to be released, right? So when we use ATP or spend it, that very literally just means we are breaking off that third phosphate, and so you wind up with a DP, right? Diphosphate, di for 2, and this third phosphate hanging off all by itself. We call it an inorganic phosphate. That's why PI. Inorganic means organic molecules have carbon. We've detached it from the carbon-containing adenosine molecule, so it's floating around by itself. When you broke that third phosphate off, the energy that was released from it, you can use that energy to fuel, right, endergonic reactions. And remember, endergonic just means requires energy, and so you're using ATP to fuel those anabolic reactions throughout a cell. That mostly means synthesis, right? So protein synthesis, lipid synthesis, making whatever the cell needs to make. Then, now that you have your ADP and phosphate that are kind of low energy and not really useful anymore, you can use exergonic reactions, which is, right, glycolysis and citric acid or Krebs cycle. you can use those exergonic reactions to add energy and tack the third phosphate back on again, getting you back to ATP. So it really is this big cycle over and over again of make your ATP, break off the phosphate, extract the energy, right? Use energy from glycolysis and Krebs cycle and cellular respiration to reattach the phosphate and start the whole cycle over again. Break it off, reattach it. Break it off, reattach it. That's all you're doing. And this is fueling all the reactions that that cell needs to carry out in order to function. Okay, so back to our big picture overview. What are we doing? We're starting off with this glucose molecule that is very high energy. It has high energy electrons. And as we go through this process, this is all oxidation of glucose, right? We're oxidizing glucose, removing its electrons. And what are we removing those electrons to do? We're giving them to NAD+, forming NADH. And so the electrons wind up on NADH, and we make a little bit of ATP along the way. Same thing in Krebs cycle. We're going to be producing lots and lots of these NADH, and also FADH2 is a similar electron carrier, right? And in the end of all of it, those NADH and FADH2s are going to give their electrons to the electron transport chain, which uses them to make even more ATP. Okay, and so all of these redox reactions, moving electrons around with NADH and FADH2, in the end, it all leads to ATP. They're just energy currency, right? The electrons are how we move energy from one place to the next. All right, so in the next video in the playlist, we're going to get into more details of glycolysis, how those reactions happen, and start building all the individual components of cellular respiration. So follow along in the playlist. Subscribe to the video, and we'll get the entire story put together for you.